This video is going to be about reading frames and how changing them can change the protein. And we're also going to talk about which direction the RNA polymerase or DNA polymerase is going to read. So let's start with reading frames. If we have a sequence that is A, T, G, A, T, C, C, A, G. So I've written them in groups of 3 here. Our first reading frame, I'll write R1, and that's in these groupings. If we were to look at reading frame 2, then we don't start with the first nucleotide. Instead, we start with the second one. So that would be TGA, T, C, C, A, G. So our groupings of 3 are now TGA, TCC, and then we have A and G left over, which perhaps the sequence continues. The point here is that the reading frames for 1 and 2 are different, and they're going to code for potentially different amino acids because we have different triplets. And if you aren't familiar with what that means, we'll talk about translation in another video. In reading frame 3, we don't look at the A and we don't look at the T, we start with the G. We start with the third nucleotide because it's the third reading frame. So this would be GAT, and then C, CA, and then G. Again, I'm grouping them in 3s because that's how they're going to be used in translation. And now we have a third set of possible amino acids using a third reading frame. The reason why we only have 3 reading frames is because if we try reading frame 4, then we don't look at the A, we don't look at the T, and we don't look at the G. We start with the 4th nucleotide, A, T, C, C, A, G, and we have the same groupings. We are missing that first triplet, but after that we're going to have the same groupings as we did for the first reading frame, so usually we only talk about reading frames 1 through 3. All right, so let's now talk about the direction of reading a gene and why we can have genes on both strands of DNA. So I'm writing out this strand 5' to 3', and we'll say ATT, GAC, CTC. And then our other strand will have the 3' end where the top strand has the 5' end, and vice versa. And we'll write in the complementary nucleotides, which would be TAA, C, T, G, G, A, G. Now, if you were to read in the context of a problem that the RNA polymerase moves in a certain direction, for example, if we say that the RNA polymerase is moving towards the right, or moving from left to right, then we have 2 pieces of information. One is this piece of information we were just given, and number 2 is that we know that the polymerase always reads 3' to 5'. That is inherent to any enzyme that reads DNA. We always read 3' to 5'. So taking these 2 pieces of information, we can say if we're reading 3' to 5' and we're moving from left to right, then we could scan left to right, and we have to start with 3', and so we must be using the bottom strand here. If we have the opposite information, and we are told that we're moving from right to left, we can still use that piece of information along with the idea that we read from 3' to 5'. And so if we start on the right side, the top strand has 3', so we know that we're using this strand. Okay, taking it one step further, what if we're not told that the polymerase moves from left to right? What if we are told that this is the promoter region right here? A little bit messy here, but anyway, we know that the promoter is always upstream of the gene, by which we mean that the promoter is going to be towards or closer to the 3' end. And then we also know that our enzymes move or read 3' to 5'. So again, if we head up here to look, we're going to move towards the 3' end. So if this is our promoter, we are reading 3' to 5'. So all right, so what if we had some information that said that the promoter is here? This is the promoter region for this specific gene. So now we could take the information. We know where the promoter is. That's our first piece of information. Our second piece of information is that, again, we know that enzymes read 3' to 5'. And then our third piece of information is going to be that we know where a promoter should be with respect to a gene. So promoter is always going to be upstream. Sorry, this is messy. Upstream of a gene. And what does that mean? It means that as you move towards the 5' end, you're going to encounter the gene. So we're going to have the 3' end, the promoter, and then the gene, and then the terminator, and the 5' end. And that's how it's going to read. So if we apply that to this diagram that we have up here, this region is our promoter region. If we're moving from 3' to 5', so when we start at 3', we move towards the 5', so we know that this top strand is the one that's going to be read for this gene in particular. Again, if we had said the promoter was on this side, then we would be talking about the opposite situation, potentially. All right, so that's it for today, but let me know if you guys have any